John and Hari. I'd like to start off with you. So, we're as I said, we're sort of in an interesting space where we're all trying to figure out where to take our pipelines, how to get through the IND, how to get into the clinic as quickly as possible. But we're also trying to work on figuring out what a good mRNA therapeutic even looks like, right? What that means. And so I'm going to focus this question firstly on linear mRNA, though folks watching working with other mRNA modalities, please note I do have a question that will be geared more towards some of the other advanced modalities. But John and Hari, and maybe we can start with John here. I'm curious to learn from you, what are the most important questions manufacturing experts should be asking about the design, right, of linear mRNA as it relates to manufacturability, and why you think those are the most important questions we should be asking as we're evaluating and working with our R&D teams today?
That's a great question. What I think is important when we look at the design of mRNA, from my perspective, is important not only to understand the expression of the mRNA, right? Because I think ultimately what's important is that the mRNA that you would design needs to be expressed appropriately, making the appropriate protein and having an important therapeutic impact, right?
Yeah.
But you need to balance that with manufacturability, right? So with manufacturability, it's important that you identify what are your key quality attributes for your product, and frankly, is it are there any impediments to the manufacturability? So what we've done is, at least at Nutcracker, what we've done is we have a part of our process, which is our lead candidate nomination process, which includes not just a look at the protein expression or the potential protein expression, but we do a manufacturability assessment as well as part of the core criteria. So you need to really look at this in an integrated way, right? There'll be some codon optimization programs that you need to establish, and I think you need to find really where that sweet spot of operability is. You can have a highly expressing process, but if you can't have appropriate yields or hitting the appropriate quality attributes, then this becomes a challenge. And conversely, of course, the manufacturability aspects sometimes are offset a bit by the protein expression. So it's really going through an optimization program and generating a fairly large data set on a platform process that enables you then to predict and forecast, both your manufacturability and your expression. And that really is, I think, the key factor for success to bring forward, rapidly bring forward lead candidate nominations, which have almost short of infinite possibilities, right, in the codon optimization sequence, and bringing the ones forward that really have, both the manufacturability aspects as well as the protein expression aspects appropriately balanced.
That's a really great answer, thinking about it from its therapeutic possibilities, right, in terms of that protein expression, but also really keeping in mind that the structure of that molecule as well, and what you have to do, and if you're going to be able to keep it in spec, right, over a long period of time and reproduce that. That's a great answer. Hari, I'd be curious to hear, is it similar in your experience as well? How do you encourage your teams to evaluate and move forward with evaluating a potential next-gen candidate?
Yeah, thanks, Anna Rose. John covered many great points. Just to add a few more, I think when it comes to manufacturing anything, you have to understand the structure of the molecule. And so, what is the cap structure that is part of this particular molecule? What is the poly A tail look like? And most important is, what is the overall length of the messenger RNA? Because the length is a key feature of this molecule. And now the good news is that the manufacturing of the mRNA is built into the discovery process. That is, you have to manufacture these in order to do the kind of experiments to get it to a development candidate. And so in some ways, you already have a sense of what it would take to manufacture that molecule already before you have to manufacture it for larger scales for clinical studies and so on. So you already have a preview of what's to come. But yeah, the key features would be the structure of the molecule, cap, tail, modifications or not, and of course the length. Those all determine the manufacturability of mRNA. And then lastly, in order to make mRNA, you have to make DNA typically as a template, and many of these features impact the manufacturability of DNA as well, and you need to consider those as well.
Yeah, that's a huge piece of it. I really like this notion that you brought up too of a lot of the questions that you have to answer about manufacturability are almost solved by exploring, right, the IVT process, creating that drug substance. So you almost get, we always talk about IVT and the manufacturing process as being more efficient, right? And in this case, I think we're seeing some of that efficiency baked in even across the CMC and the drug discovery world, right? They're very closely intertwined.